Long, multinucleate and full of actomyosin filaments, it's hardly surprising that the intracellular organisation of skeletal muscle fibres bears little resemblance to that of regular proliferating cells. Muscle microtubules, for example, are organised into a grid-like network instead of forming asters that radiate outwards from a clear microtubule organising centre. Defects in this network may underlie the pathology of Duchenne muscular dystrophy, but Evelyn Ralston from the NIH in Bethesda, Maryland, says that the organisation of muscle microtubules is poorly understood.
When we did immunofluorescence staining to look at the distribution of EB proteins, which are supposed to be at the plus ends of the microtubules, we would see them along the microtubules, not necessarily at the end. So there were a number of observations which we could not truly explain based on what we know of microtubules in other cells. And we thought at some point that the only way to understand how these microtubules are organized and formed was to look at them in live cells and, if possible, in live animals.
Ralston and colleagues, led by Sarah Oddoux and Christine Zahl, therefore observed isolated muscle fibres expressing a GFP-tagged version of the microtubule plus N marker EB3.
In principle, EB3-GFP is only bound to dynamic microtubules. So we were extremely surprised and excited really when we saw that all the microtubules that we see by immunofluorescence seem to be actually growing and dynamic. So we saw a lot of dots moving, mostly with the same directionality as the lattice that we see by immunofluorescence.
The EB3 puncture moved left, right, up and down at average growth rates of about 9 microns per minute. Many of the dynamic microtubules seem to be nucleated from intersection sites throughout the lattice. Oddoux et al. Confirmed their observations in vivo, using intravital microscopy to visualize EB3 dynamics in intact living muscles. The researchers then set out to visualize the entire microtubule system by imaging GFP tubulin.
When we started injecting and expressing GFP tubulin in the muscles, we had the feeling suddenly that we saw a very static network. It seemed to be persistent. We recorded 4 minutes and we saw very little change. There were some areas, we call them pockets, where you do have dynamic microtubules growing, shrinking, but truly we saw a more static network than what we saw with EB3-GFP. So then we needed to reconcile these different observations.
Photobleaching experiments revealed that most of the microtubule dynamics seen with GFP tubulin could be attributed to microtubule growth. In addition, to reconcile the different behaviours of EB3 and tubulin, Oddoux et al. Co-expressed both markers in muscle fibres and saw EB3 puncture moving along static tubulin-labelled tracks.
So that gave us the feeling that we must have bundles of microtubules, and we have dynamic microtubules growing on pre-existing microtubules, which could either be stable or could themselves be dynamic. But anyway, this told us that we need to have small bundles of microtubules. And they have to be antiparallel because we can see EB3 moving in both directions on what appears to be the same microtubule.
Super-resolution microscopy revealed that the microtubule lattice was indeed composed of bundles of 2 to 4 microtubules. But where do these microtubules come from? It's known that muscle microtubules can be nucleated from the nuclear envelope, but Oddoux et al. Had also observed microtubules growing from the intersections of the lattice, where small fragments of the Golgi complex, known as Golgi elements, can also be found.
So we treated the fibers to completely depolymerize the microtubules, and then we let them repolymerize. And we noticed that at very short times, we start seeing very short microtubules around the Golgi elements. And a few minutes later, you see very clear asters sticking out of the Golgi elements. So that indicated that the nucleation of the microtubules is done in association with the Golgi elements.
Two proteins commonly found at microtubule organizing centers, gamma-tubulin and pericentrin, localized to the Golgi elements of muscle fibers, and live imaging showed that the Golgi fragments are largely static, suggesting that they form the microtubule lattice by nucleating microtubules in different directions. But how are the microtubules arranged into an orthogonal grid? Ralston and colleagues think that the microtubules might be guided by dystrophin, the microtubule-associated protein that is lost in patients with Duchenne muscular dystrophy.
It has been shown a few years ago that the microtubules in the MDX mouse, the mouse model for Duchenne muscular dystrophy, are not organized orthogonally as they are in wild-type mouse muscle. They tend to be more randomly distributed. Now we are looking at the MDX fibers, and we hope to really understand how the disorganized microtubules are formed in comparison to the wild type.
In the meantime, you can learn more about the organization and dynamics of muscle microtubules nucleated from Golgi elements in the paper by Oddoux et al., published in the October 28, 2013 issue of the Journal of Cell Biology.